I just wanted to begin by expressing my sincere gratitude to the Linda Hall Library for supporting my research. Being a Linda Hall Library Research Fellow has given me the opportunity to access a breathtaking collection of texts in the history of comparative anatomy and physiology. Everyone here has been so warm and friendly, making my research both productive and immensely enjoyable. I am particularly thankful to Cindy Rogers, Bruce Bradley, Bill Ashworth, and Nancy Officer. Their help and advice in the rare books room has been invaluable. So, since the focus of my discussion will be investigating the nature of the concepts of homology and analogy, I hesitate to begin with a definition of these. So instead, I'm just going to give you some working definitions. So, homology. Homology tends to be understood as a similarity or sameness of structure and orientation in a wide variety of different organisms due to modification or deviation from a shared origin. So my arm, a whale's pectoral fin, the wing of a bat, or the forelimb of a horse. Analogy, on the other hand, can be understood as similarity of function due to similar purpose or goal or similar solution to similar environments, such as a butterfly's wing, a hummingbird wing, or a bat wing. So my discussion centers on Richard Owen. Richard Owen was a Victorian comparative anatomist who was a contemporary of Darwin. Owen is perhaps best known for coining the word dinosaur, curating the Hunterian Museum in London, cataloguing its 13,000 human and animal specimens, and setting up the British Natural History Museum in 1881. Owen's contribution to comparative anatomy grew out of his interactions with these collections of specimens and led to his being recipient of many specimens from Darwin's voyage on the HMS Beagle, and led also to his meeting Cuvier in 1830. But it is actually not these contributions to natural history that I will be discussing. As a philosopher of biology and with a focus on classification and the nature of kinds, my interest is in analyzing the comparative concepts Owen articulated and understanding their use within comparative anatomy. The observation that different animals share strikingly similar features among some of their traits is not new, and it didn't originate with Darwin. A record of comparative anatomy and physiology and the identification of sameness of form in different animals and in the different parts of animals, such as iterations of structure, segmentation, dorsal and ventral polarity, has been observed and recorded since Vesalius in 1543. In the 16th and 17th centuries, some of these observed resemblances were thought to be accidental similarities. Others were believed to be the result of deliberate design for a specific purpose. For instance, many aquatic animals shared traits such as fins, while other animals occupying aerial environments tended to have wings. Animals as diverse as blowflies, manatees, octopi, and crocodiles all had eyes. And the bone configurations of bats, whales, and humans' forelimbs seemed to be arranged in a similar way to one another. If the similarities of these traits was not accidental, then their presence in a wide variety of animals required some kind of explanation, explanations that identified the reason for these similarities. Whereas some traits, like the paired fins of aquatic mammals, appeared to share a similar function, enabling the organisms to stabilize and balance itself within an aquatic environment, others, like the forelimbs of bats, whales, and humans, served completely different functions. If these anatomical structures appear to be the same structure in different animals, even though they do not share similar functions and are not accidental, what then is the nature of their similarity? The observed similarities among these diverse structures were due to their similar positional arrangement, similar arrangement of bones of the forelimbs and connections to adjacent bones of the skeleton. So rather than functional similarity, the reason for their similarity was their shared structural configuration. This view led to the classical account of the comparative functional and structural similarities among organisms. In these, the reason for their similarity was frequently given in terms of the reflection of an ideal form, so references to Platonic forms or Goethe's idealist Naturphilosophie. The resemblance of individual traits of different animals to one another was explained in terms of their common cause. Both traits were the approximated reflections of an ideal form or trait. Homology is then a particularly and notoriously elusive concept to pin down. There has been, and continues to be, sustained debate over the meaning of homology, the nature of correspondence, and the units of comparison. The focus of this talk then centers on analyzing the nature of the comparative approach and the epistemological practice of homologizing. This fits within my larger project, critically investigating the conceptual consequences of the analogy-homology distinction by considering its conceptualization and use in both historical and current research that include both pre-Darwinian notions of homology as well as those that are forming within developmental biology and the extension of the modern synthesis. I suggest that these debates surrounding the notions of homology and analogy persist and appear at times to be iterations of earlier debates. Because of this, I will suggest that research into the historical discussion over homology may not only be better to elucidate past conceptual problems, but could also offer solutions to problems surrounding the use and understanding of the conception of homology in new areas of biological research. So why is this all important? That's the big question. Advances and innovations in biological research and technology affect our knowledge and understanding of the kinds of things that populate the natural world. And that we understand these in large part through our understanding of the nature and cause of their similarities and differences. So how homology is understood and used changes how we distinguish and classify the world's inhabitants. This will not only impact theoretical discussions within philosophy of biology, its effect may extend to all fields using these comparative concepts, from systems ecology to synthetic biotechnology. So my talk then is both backward-looking and forward-looking. I begin then with a survey of the early classical conceptions, and following this, I will provide an account of the nature of homologizing as an activity that includes a rough sketch of the concept in use. In Vesalius's 1543 Structure of the Human Body, there were representations of serial repetition of form and structure of bones, muscles, and general parts, indicated with the use of the same Greek letters. Although the theoretical implications of the comparison may not have been understood by Vesalius, there are images that suggest the comparison was being used. For example, the use of animals as comparative epistemological subjects are present in a number of places. They are representations of a dog and a human skull, and a dissection of a pig that Vesalius featured in his historiated capitals. Much of Vesalius's research and textual information that we have shows that he relies on clear knowledge taken from dissections of animals. Underlying the use of dissection of other organisms as stand-ins for humans is an unstated assumption that there is a unity of form, or at least of knowledge of orientation. That pigs and dogs were dissected strongly suggests at least a comparative approach in practice to the objects of investigation. Although the study of anatomy has been cited as originating back to Aristotle's On the Part of Animals in 350 BCE by later investigators such as Vesalius, the earliest record of explicit structural equivalence between features of different animals can be found in Bellon in his Anatomical investigations in 1551. Here we can see evidence of early comparative approaches both in 1551 and in 1553. Probably, though, the most frequently cited is in 1555. These detailed sketches compared the positional arrangement of the bones of a human with those of a bird. What was particularly significant in these sketches was how Bellon arranged the bones of the bird and the bones of the human skeleton in the same upright position. Actually, they're on little hooks, you can see. So a bit macabre there. But so what was the particular significance then? Is they're aligned to look in the same upright position. So the skeleton of the bird was in a quite unnatural position that mirrored the normal upright position of the human skeleton. So its head, neck, and spinal column form a straight line, and the forelimbs hung down on either side of the body in a ventrally splayed position. So rather than picturing the bones of the bird in their natural position arranged in a living bird, the side-by-side upright alignment of both the bird and the human skeleton emphasized the positional equivalence of the corresponding topology of the bones in each animal and the similar skeletal relationships. Following Vesalius and Bellon, Rooney in 1603 also uses corresponding letters to muscles and bones, especially in his vertebrae. He likes to hang them on strings, you can see that. But he also has quite significant portrayals of what could be later understood as serial homology in terms of the ribs. Also in Grew, Grew compares cross-sections of plant stems, that's at the top, and other comparative organs in the 17th century. Grew also has a striking series of images of descriptions of comparative organs and an ordering of comparative parts. My favorite is this one. In 1681, it's a series of guts of various animals, all lined up and oriented in the same way, just right next to each other. So to indicate a comparison of structure and explanations of functional affinities and differences. Perhaps the most influential discussion on comparative anatomy prior to Owen's formal distinction between homology and analogy that I will get to in a bit occurred with the debates between Cuvier and the idealist Geoffroy. And this occurred in around the early 1830s in Paris. Cuvier and Geoffroy held different views on what they believed to be the significant relationships among corresponding traits in different animals. A caricature of this position often given is that Cuvier argued that similar functions are the cause of structural similarities, whereas Geoffroy argued that structural similarity is the cause of the similarities of function. However, this perfunctory description of their debate obscures many conceptual nuances of the two views. To fully understand the basis for these competing views, it will be necessary to consider the theoretical basis of their individual positions. I begin with Cuvier. Cuvier's theory of embranchements claimed that there were 4 different basic body plans of animal structure: vertebrates, such as bony fish, birds, and humans; articulates, lobsters, horseshoe crabs, and dung beetles; radiates, so that would be sponges, sea anemones, and starfish; and mollusks, snails, octopi, and cuttlefish. In positioning these 4 body plans, Cuvier's theory took certain sets of body parts, features, or traits to be found together in certain animals. Traits could not be found just in any arrangement or in any kind of admixture, but only within the constraints of one of the 4 body plans. So Cuvier's embranchements were not ideal types at all. They were actual combinations of traits that were found together in particular animals. These groups of traits acted in cooperation with other traits of the organism towards a particular goal or purpose. The upshot of Cuvier's theory of embranchements was that only sets of traits which act together in this way can actually be found in nature. This was because the structure and arrangement of traits was dependent on the environment and conditions for the sake of which they were formed in the animal. Put another way, the traits of animals correlated with the conditions required for the organism's existence within a particular environment. Cuvier understood the organism's functional unity in terms of its interdependence of traits towards a telos. In this respect, he was influenced by Kant's view of organisms as natural purposes. By conceiving of homology teleologically, Cuvier understood the similarity of parts between two different animals as due to the similarity of their functional goals or purposes. For him, the animal's mode of living determined the arrangement, position, and relationships of its appendages, organs, and other features. For instance, if the animal lived in the deep sea, it would require a soft invertebrate body plan so as to withstand great pressures and suctioned tentacles for easily grasping prey in low light. Thus, a body plan characteristic of mollusks. The purpose for which these traits are used by deep-sea living animals, such as an octopus, might include the ability to dive to great depths, collapse its body to fit through small openings between rocks, capture slippery prey with little or no light, quick propulsion through an aquatic environment, and the ability to attach itself to various surfaces. These are what determine its structure: a soft invertebrate body with 8 leg-like appendages with suckers on the underside attaching to the base of the head surrounding the mouth. In contrast to Cuvier's embranchements, Geoffroy believed that there was only one body plan. It was common to all animals. All animals and all organs and all appendages and all other traits were based upon the same shared universal plan. For Geoffroy, the organization of animal form was the differential expression of one generalized, abstract, universal type. Homological correspondence, then, between the same organs in different animals could be explained as the result of animals sharing the same universal plan. Two organs are the same if they are located in the same position, in bodies of two different animals being compared. Any variations, then, among these organs were the independent modifications of each animal on the universal body plan. There might be an infinite variety of modifications observed among different animals. Geoffroy expected wide variations in form and function of homologous organs. What mattered in the comparison of homologs was only that their position remained constant. So he said that an organ can be deteriorated, atrophied, annihilated, but never transposed. The centrality of positional continuity formed the foundation of Geoffroy's theory of homology, the keystone of which was his principle of connections. So whereas Cuvier's notion of homology focused on whether interdependent structures shared a common functional goal or purpose in different animals, Geoffroy's reliance on the abstracted notion of a universal body plan meant that his understanding of homologs in organs of different animals depended on their relative topological position. So Cuvier's body plans were discrete and fixed. There was no transformations and no intermediates between the 4 embranchements. For Geoffroy's, the universal body plan was almost infinitely flexible, producing a continuum of intermediate transformations and modifications of the universal body plan. Shortly after the debates between Geoffroy and Cuvier, Owen now comes into the picture, used notions very similar to both Geoffroy and Cuvier's in 1832 and 1840. In mostly the style of Cuvier and Geoffroy, Owen's use of analogy actually was meant to be homology. At this time, he had taken on the role of assistant curator of the Hunterian Museum. John Hunter in 1763 started the Hunterian Museum and used its contents to explain the order and form. Actually, the whole organization of the museum is organized to display or to exemplify the natural order of the world. So from 1826 to about 1830, Owen catalogued all of the 13,000 specimens of the Hunterian Museum. But of the many of these specimens, there's one that he was particularly intrigued by, and this was the pearly nautilus. He was so intrigued, he actually wrote an entire memoir about it. So in 1832, Owen's memoir of the pearly nautilus includes use of analogy and analogous on almost every other page. It's quite striking, especially since most people don't actually have a look at this when then researching Owen. He also used precisely analogous, and that seems to, it appears to refer to homology or some sort of continuity of structure. But he also talks about affinity and parallelism and non-analogy. But he uses these terms in a less consistent fashion. So this is actually Owen with his prized nautilus. Quite sweet. So Owen suggests that his investigations of the nautilus led him to conclude that there was not a simple and unbroken series or plan of composition or a principle of unity, so very Cuvierian. But then on page 15, he seems to offer a very Geoffroyan characteristic, saying the existence of a plan, a general plan of organization is indisputable. So he kind of wants to have his cake and eat it, you know? So have a bit of Cuvier, a bit of Geoffroy. So here's where our hero comes into the story, depending on which side of the Owen-Darwin kind of debates you're on. But so during the voyages of the HMS Beagle, Owen was the recipient of many of Darwin's fossils. And actually, he was the author of the 1839 Fossil Mammalia, which is part of that. In this, he uses analogy to describe what we know as homology, to describe many of the fossils that he was given by Darwin. It's perhaps the reason why it's not the 1839 version of his distinction between homology and analogy that people usually talk about with regard to this formal distinction. The formal distinction appears in 1843 and 1848. This is where he recognizes and actually gives definitions for functional similarity, which he calls analogy, and structural similarity, which he calls homology. So he identifies the cause of similarity of analogous organismal traits in terms of the common functions they serve. And this is at the top corner there. Analog, then, is a part or organ in one animal which has the same function as another part or organ in a different animal. Unlike analogy, Owen's definition of homology contained no such causal explanation. Homologue, he refers to as being the same organ in different animals under every variety of form and function. Perhaps Owen's 1843 and 1848 can be seen as a codification of a view prefigured in 1832 and 1839. Owen believed that although the Cuvier and Geoffroy debates drew attention to the importance of the concepts of homology and analogy to comparative anatomy, they also beclouded their meaning. Owen's aim was to provide a clear and consistent definition of homology and analogy, something that had not been done either by himself or other people previously. Despite these criticisms, Owen makes explicit use of Geoffroy's principle of connections in distinguishing homology and analogy. Owen's definition of homologues as the same organs is intended to capture their topological orientation and structural correspondence in different organisms. Homologous organs, such as the wing of a falcon and the foreleg of an ox, were identified in terms of their corresponding topological location within the falcon and within the ox in relation to other features, so the position of the humerus and its attachment by ligaments to the ulna and radii, claws, and hooves of each animal. In contrast, the biological concept of analogy was defined in terms of the functional similarity of two organs in different organisms. For instance, the wings of a blowfly are analogous to the wings of a hummingbird. These are identified as such because of their similarity in terms of their functional role in enabling the animal to maneuver itself around an aerial environment. Owen drew inspiration from Cuvier's theory of embranchements, as well as Geoffroy's principle of connections. But Owen did not believe that Cuvier's teleological point of view sufficiently explained the similar topological correspondence of organs and positions across a variety of animals. So he augmented it with Geoffroy's notion of the universal body plan, what Owen actually called the archetype. So this is actually pretty amazing plate. The top skeleton is what he refers to as the vertebrate archetype. And then he shows the similarity of all the other organisms in terms of its similarity to the archetype. He also has the ideal typical vertebra also here. So for Owen, the archetype then, this is the full picture here. So for Owen, the archetype then referred to a common plan or a structural arrangement. Owen believed that there was an ideal common plan or structure for all vertebrates. And so apart from distinguishing the distinction between homology and analogy, Owen also provided the first account of another 3 different kinds of homological relationships between compared organs of organisms, one of which was general homology. So general homology was the comparison of 2 or more organs as similar, where their similarity was caused by the similarity in the same archetype. So this is his view of general homology. This was distinguished between 2 other kinds of homology, special homology and serial homology. So special homology was defined as the comparison of same organs within 2 or more different organisms. The relationship does not include the comparison of the archetype, but consists just in the correspondence of parts, as determined by their relative position or connections. The third kind, serial homology, refers to the different parts or organs within the same animal. So these parts are thought to possess the same structure. So vertebra or leaves of a plant or even hairs on one's head. So a repetition of structure within the same animal. Although intended to identify the identity of 2 organs, Owen's notion of sameness employed was not meant to apply any kind of material identity of 2 organs. After Owen formalized the definition of homology and analogy, attempts were made to provide a causal explanation of similarity of homologs, or of homologies, and this became more widespread. Comparative embryological studies of the morphological features of many organisms' early anatomy led both Haeckel and Darwin to reconceive Owen's concept of homology in terms of the historical continuity of traits over generations. However, whereas Haeckel's historical notion was based on the theory of recapitulation, Darwin's was based on the theory of descent with modification. Darwin's theory of descent with modification provided a causal explanation of Owen's homology and replaced the classical notion of the similarity of corresponding structures to an ideal form or archetype with that of a common progenitor. Homologs were the result of descent with modification from a common ancestor. And so the study of homology was the comparison of traits of organisms in terms of their similarity or correspondence to a shared ancestor. The wing of a bat and the arm of a human are homologous to each other because these traits were conserved over many generations of organisms and descended with modification from the same four-limb structure in the nearest or most recent common ancestor of both bats and humans. However, the explanation of sameness of these four limbs, the bat's wing and the human's arm, is not contained in the identification of their similar bone structure. It's not their structural correspondence per se, but their historical continuity, the inheritance of a certain feature with modification from the same ancestor, which is then conserved over generations. The conception of homology as correspondence due to shared ancestry may initially seem an unambiguously straightforward conception of biological relatedness. However, little credence should be given to this initial impression. Darwin was very keen to distance himself from the idealism of Owen and Geoffroy and their particular use of the archetype. Avoiding this idealist notion meant that in order to integrate Owen's concept of homology into his theory of evolution, Darwin needed to make some alterations to its meaning and reference. Although Owen's special homology made no reference to an archetype, it also provided no causal explanation to the topological correspondence of organs in different animals. It was merely a description of similar correspondence. The only kind of homology characterized by Owen that included a causal explanation within its definition was that of general homology. The similarity of two corresponding organs in different animals to each other was caused by their similarity to the same organ in the archetype. Although Darwin sought to avoid the commitments to idealism, which he does not rely on with regard to any other kind of format, he does not rely on Owen's special homology. Owen's special homology was the one that did not rely on the archetype. Instead, Darwin uses Owen's general homology, which is the one that does rely on the archetype. Looking at the role homology plays in Darwin's theory reveals why. Darwin does not shy away from actually using the term archetype as well, as my little arrow shows. So, just in case you missed it. So for Darwin, homology is not just a description. It's a causal explanation. It's obviously what he was going for. Organs correspond to each other because they both correspond to the same common cause, a shared ancestor. Darwin's concept makes use of the same form of explanation as Owen's general homology, but interprets the archetype differently in terms of the historic notion of shared common ancestry, rather than the idealist notion of an archetype. And I'll just read this because, just in case it's kind of hard to read there. So suppose that the ancient progenitor, the archetype, as it may be called, of all mammals, had its limbs constructed on the existing general pattern. For whatever purpose they served, we can at once perceive the plain significance of the homologous construction of the limbs throughout the whole class. And that's in Darwin's 1859. In that passage, Darwin does not shy away from using Owen's concept of the archetype explicitly in his historical conception of homology. The historical notion of Darwin's ancient progenitor replaces Owen's idealist archetype. According to Darwin's historical notion of homology, the traits of two different organisms are not homologous because of their shared function or form, which may differ quite greatly, and not because of their position or orientation. These traits are homologous because they have been inherited and modified from the same trait in an ancestor common to them both. Vocal critics of the classical views of homology focused on the inability of these views to account for developmental perturbations, phenotypic modifications, environmental influences, and the different mechanisms by which homologous traits were inherited. Rather than focusing exclusively on the phylogeny of organisms or simply just black boxing development, as was the fashion for quite some time, Gavin de Beer in 1971 reasserted the importance of the role of organismal ontogeneticist. What was absent in the historical conception of homology was a clear understanding of the causal roots of inheritance and modification leading to morphological outcomes from common ancestral forms. Remedying this omission required careful consideration of the role of development in the construction of these morphological traits in individual organisms. De Beer identifies particular limitations of these views. He objects to the practice of using corresponding developmental processes or similar cellular tissues to justify morphological homology. De Beer argues that problems emerge when one tries to identify similar developmental processes or cellular substrata as evidence for morphological corresponding homologs. He reveals these problems using a number of examples. Relying on the same underlying cells, the similar position of these cells in the embryo, or corresponding parts of the zygote from which the different structures are formed, does not guarantee the homology of the morphological structures which are constructed from these. Relying on the same cell position area of the zygote or the same genes as evidence for homology encourages the erroneous assumption that homology at one level, the level of morphological traits, is caused by the homology at another level, the same cells or genes. Revealing the falsity of this assumption were de Beer's studies of the incongruities between the corresponding similarities of morphological traits, developmental patterns, and his studies on Drosophila, especially using what are called eyeless mutants. Observing eye development in fruit flies with this eyeless mutation, de Beer found that while some with this mutation do not develop eyes, other eyeless mutations actually develop fully functioning eyes that are identical, morphologically speaking, with the wild type. He realized that there was actually a set of genes in some of the mutants, which he referred to as modifier genes, that when present resulted in the eyeless mutant actually developing fully functional eyes. His observations were startling, not only because the mutant eyeless fruit flies actually developed eyes, but because their eye development was through completely different developmental routes. De Beer's investigation of the eyeless fruit flies calls into question the assumption that the gross morphological similarities or correspondences between organisms were solely caused by the same genetic or developmental mechanisms. So ever since de Beer's studies in developmental biology, people have been continuing to investigate these in terms of many of the Hox genes, PAX6, eyeless, there's many, many others. So ever since de Beer revealed that homology at one level does not actually imply homology at another level of organization, there has been constant debate over which is the real level of homology. What is the source of homology? Is it genetic? Is it cellular? Is it developmental? Is it morphological? This debate betrays a complete misunderstanding of de Beer's findings. There's no privileged level of organization which causally is responsible for homology at all levels. It is instead a relationship that can only hold independently of a number of different levels. Discussing the role of the Hox genes that are involved in the regulation of patterns of anatomical development and morphogenesis in animals, such as fungi and plants as well, developmental biologist Brian Hall notes, Homology is studied and can be defined at all levels of biological organization, from molecules through genes, cells, organs, embryos, organisms, populations, communities, behavior, and even biogeographical regions. So homologies at different levels of organization are independent of one another: developmental pathways, morphological traits, regulatory networks, or genetic sequences. So this means that each one of these could be potentially a unit of comparison. Well, this may, with this, leads us back to the original question of Owen and others. What is the nature of homology? That's what I set out to kind of discuss. What is the nature of thinking about comparative anatomy and the nature of biological identity? What does it mean to say sameness of form? Where to start? What is common with the comparative approaches to Hunter, Darwin, Owen, as well as Nehemiah Grew in his lovely catalogue of guts, was the collection itself. Investigating the collection itself was used to ascertain the sameness of form in different organisms. The collection had an epistemological role in the development of the archetype, the development of the distinction between homology and analogy. As an assembly of various parts of various animals, these were en masse observed, and from these, generalities of the specific specimens were found. My suggestion is that comparative thinking in general and homology thinking in particular appears to be something that is best understood in practice. Instead of starting with the theory and considering implications for scientific practice as coming from the understanding of theory, I suggest taking seriously the significance of scientific practice and then asking about the implications for theory about or defining homology. This would provide an understanding of homology in use rather than defining homology and analogy in theory and then seeing what happens in practice. This move from practice to theory not only takes the role of scientific practice seriously, it inverts the epistemic methodology that is usually used to analyze scientific concepts. In my remaining time, I'm going to give a very, very rough sketch of what it is that I'm suggesting. This is very, very, very much a work in progress. Okay, so I would like comments at the end. My suggestion is to get to grips with the nature of homology and analogy. What needs to be the focus of critical inquiry is actually the act of homologizing. My suggestion is that homologizing as a practice could be conceived of in terms of the tracking of things over time and across different organisms. It tracks an identity relationship, but identity not of substance, but of orientation or position. The philosophers. So this could be loosely understood as kind of sortal tracking, using a particular notion of sortalizing, a bit like E.J. Lowe. A sortal concept is a concept of individuation of a particular kind. A sortal provides a way of counting parts of a particular kind. That is, a sortal specifies whether two or more parts are identical or non-identical, and provides a way to signify what it is that is being referred to in terms of its kind, and the letters are indicators that can be referred to in something like the legend as identifying a particular kind of part or organ. So it's a forelimb or a lung or a protein. This goes back to the observations I made at the beginning of the talk about Vesalius, Rooney, and Grew, and their use of letters as signifiers of parts. For these letters to actually indicate parts, their use must have been underpinned by an assumption of identity of things being signified with the letters to indicate the same parts in different organisms or in two different locations within the same organism, as in serial homology. And this seems to be something that actually Owen himself was using. So perhaps the nature of homology and analogy can be characterized as a series of sortalizing activities of homologizing and analogizing. The verb sortalizing is meant to refer to kind of a partitioning or chunking up of wholes into parts, which are then named. Criteria of individuation and identity in practice in the articulation of the name with the meaning. So one way of understanding the nature of homology and analogy is inherently underpinned in their active use and signification of the parts being homologized and analogized. These activities are the epistemic movements or processes of thought that involve analyzing, enumerating, and solving. My hope is that this practice approach would allow for different movements of thought to be required for identifying identity of form or process at different levels in developmental biology, embryology, synthetic biology, genetics, and ethology, and thus, hopefully, maybe, possibly, attempting to solve de Beer's problem. So this is just my works cited, not the complete. And I just wanted to thank everybody, and you can follow me on Twitter.
We're broadcasting live over the internet at livestream.com. So we hope that in the future you're able to attend our events in person. But if you're not able to, or if you know a friend or family member wherever in the world, they'll be able to watch most, if not all, of our lectures live. And of course, we're also videotaping. You can go to lindahall.org. About a week after each lecture, and the video will be up on our website. With that, I think we're ready. Raise your hand if you have any questions. Yes, sir. Just be sure to hold it close to your mouth.
Where were the intelligent design advocates when all this was going on?
I'm sure they were in the mix, but that wasn't the focus of my research trajectory. So yes. Can I dodge that one? Okay.
Wasn't Cuvier basically from the far right religious aspect when he applies his information to this concept?
I don't know, actually. I'm not, so that would be something to look into. I haven't looked into their religious or political kind of where they were coming from. So I'm hoping that I'm not going to have to dodge all questions. I mean, that would be something that I think would be a project that would be amenable to someone who is interested in actually doing a historical discussion of that in terms of the history of religion, or within kind of more socio-political discussions. I was just interested in the concepts. So kind of true to form, philosophers try to only talk about the philosophical concepts without thinking so much about the sociological implications. I know that causes some problems.
I think it's actually pretty incidental to the whole debate and the way that Professor Kendig is discussing it, but I think Cuvier was a leading Protestant figure in France during the period, but the debate works itself out on scientific terms, and I only happen to know this because I'm reading a book by Hans-Peter Ryle, and it's called Vitalizing Nature, and it's discussed in that text. So that's a point of reference, but it's not substantive to the discussion as it's being presented, I don't think.
Dr. Kendig, in the hopes of better understanding your homologous method, how would homology, what would it bring to the pursuit to the flavoring, the better understanding, what have you, of the pursuit of common ancestor between us and Neanderthals, or the whole pursuit of common ancestors and fleshing out the entire hominid tree, or is that applicable?
I think it would help insofar as we would understand actually what we are looking at, what we're doing when we are classifying the world. What is the process? So I would actually say probably in resolving the phylogeny, it might not cause a great deal of difference at all, but we would know what we were doing when we were actually classifying things.
Concerning the very end of your talk where you were discussing the sortalizing, I'm interested in what sounded like you were getting at the issues about ontological priority of wholes versus parts, and that the sortalizing seems to be working in this way, that the whole is ontologically prior to the parts, but the practice of sortalizing then seems to reify the parts as distinct from the wholes, which would then break off them conceptually and ontologically. So if you could say a little bit about that, if whether I'm confused about that relationship, or you could highlight it a little bit more.
Okay, so I should probably say a little bit more about the general project. So thinking about this activity of sortalizing, I'm taking quite a kind of neo-Kantian approach. So in investigating the world, there's kind of a precondition for understanding what it is that we're looking at. And to do that, part of what I'm suggesting is that we have this chunking up way of approaching the world. So it's a non-reductive account, which takes wholes, and we understand things in terms of their parthood through this process. So it's pushing together the ontology and epistemology of experience. Is that helpful?
I have a similar question to that. I was curious about the sort of metaphysical implications, in the sense that this homologizing or sortalizing in practice, do you take it to be actually latching onto anything, or is it constitutive of the sort of homologies that there are? So do you think basically there are sort of ontological categories that sortalizing in practice gets at, or is the actual sortalizing, does it constitute the actual homological relations that you find? Maybe you already answered that a little bit, talking about the neo-Kantian view, but I'd be curious to hear a little bit more about that.
Thank you for that. So, yeah, I mean, it's constitutive, absolutely. So our engagement with the world, our understanding of the world is through our activities. So it does take this rather different approach from practice and engagement with the world to understanding the concepts that we use, rather than vice versa. So it's constitutive in this neo-Kantian way. So like a frame that through which we understand the world, we cannot otherwise. I mean, we kind of are ineluctably drawn to chunking up the world. That's the way we kind of understand it.
Historically, how did Owen and Cuvier and Geoffroy argue with one another about whose position was the better one? Was there data they appealed to in the particular collections? Were they just conceptual debates? And then secondly, to your view, are there ways to say that this way of sorting the world up is better than this other way of sorting the world up? And how would one argue that one is better than another if that's a relevant question?
Okay. Reference was being made to collections, especially in and I mean, Owen was explicitly doing it, as were both Geoffroy and Cuvier. They had a series of discussions about mollusks. Cuvier actually came to visit in 1830 to the Hunterian Museum. That's where he initially met up with Owen. So they were very much kind of referencing these organisms and debating what eventually became kind of like the source of our understanding of homology and analogy. As far as weighing up better and worse approaches, it's always the pluralist's problem. Okay, me kind of like being a pluralist. So if there is homology on various levels, I think the question you're pushing is, well, either there's one that's better and one that's worse, or all of them okay, or all of them equally good. And I would think that an activity approach would at least avoid some of the kind of more problematic notions, the ones that don't seem to kind of fit the way the world is. That's a very bit half-baked, sorry. But I think at this point in time, I definitely am at the beginning stages, and so that's something I definitely want to go into more research about. Thanks.
Thank you, Dr. Kendig, for the wonderful lecture.